Hi, in this video, we are just going to take a look at some points of emphasis from the nucleic acids video. So again, um, as you'll know, nucleic acids, we have encountered them during the third quarter. So you have DNA and RNA primarily, and uh, and they are responsible for the storage of genetic information and also the expression of this information. And this is the basic layout of a nucleotide, which are the monomers of nucleic acid. So I guess that is the first point of emphasis, that in RNA and DNA, you need a nucleotide, and this nucleotide, you stack them together to form RNA or DNA. And again, the nucleotide has different variations. The nitrogenous bases, for example, it could be adenine, thymine, cytosine, or guanine for DNA. But for RNA, it could be adenine, cytosine, guanine, and uracil, right? And then for the sugar, obviously deoxyribose for DNA and ribose for RNA. But again, same pattern. You stack together nucleotides to form a strand, right? And this is the second point of emphasis, which is the type of bond formed in this biomolecule. And that type of bond is what we call a phosphodiester bond. Phosphodiester, from the word phospho, that's because of the phosphate group. And then diester, that means you have two esters at either side. Okay, so think of it as phosphorus joining two ester groups or two ester linkages. So that's why we call it a phosphodiester bond. And again, that makes up the sugar phosphate backbone. And again, for the DNA, you have two strands, which you then also bond together through hydrogen bonds with the familiar A to T, C to G bond. Or again, for RNA later on, that's T and U. When you have the mRNA pair with the DNA strand during the transcription part. But again, that's the idea. That is the difference between RNA and DNA. And again, for RNA, is just for the expression. So when you read the DNA, you use the mRNA. When you transport all the amino acids so that you can string together proteins, you use the tRNA. And even in the ribosome itself, ribosome contains ribosomal RNA. So those are the three uses of RNA. For DNA, it's really long-term storage of the genetic traits. This can go on for from generation from one generation to the second generation. So when you have an offspring, basically the sperm of the father contains half of its genes, half of the father's genes, and the egg cell of the mother contains half of the mother's genes, and therefore you combine them to form another human being, right? But aside from DNA and RNA, there are again two. This is the third point of emphasis. There are two other important uh, biomolecules that are based off of nucleotides. So you can uh, strictly say that this is also a nucleic acid molecule, or you could say it is a modified nucleotide, right? And again, in science, you don't have a straightforward way of definition, right? Because you could argue that sir, this is not a nucleic acid, because a nucleic acid is a chain of nucleotides. This is not a chain of nucleotides. This is just one nucleotide. But you have to agree that this is more related to DNA and RNA than anything else, because if you think about it, it's really just a nucleotide modified to have two phosphate groups, right? So that's what we call the adenosine triphosphate. So we have three phosphate groups here, and again, you break up that third bond to release energy to have you to have what we call ADP, and later on, this ADP will be recharged, right? And form ATP back by the um, mitochondrion. And again, that serves as like a method of sending energy to some parts of the cell. And again, this is the fourth point of emphasis. that again, in terms of relationship of how your cells work, your mitochondrion or mitochondria in plural will be the one creating ATP, but those ATP will be used in different parts of the cell. So, for example, if the ATP is over here, if the mitochondrion is over here, and what uses it is um, the steroidal gate in the cell membrane, for example, to open up or close up that gate so that uh, steroid-based compounds can enter the cell. To move that gate, it needs energy, but the mitochondrion is far away from that gate. So unlike in our modern society where we have the power plant and then we have wires that deliver the energy directly to the specific place that uses it, in their case they don't. What they do is the mitochondrion produces ATP, and that ATP is transported to that gate. And then when the gate now needs energy, it's when they would break up the third bond and form ADP. And again, as they break up the third bond, it releases the energy, and the energy is captured by the gate to open up the gate or close it, whatever the case is. But think of it as in modern society we have wires, but if there would be an equivalent for that in modern society, that would be like batteries, right? So a power plant produces batteries, and that battery is shipped to wherever you want to consume energy, and then you use that battery, and that use part is the analogy for breaking the third bond and using the energy that you can get from that. Then after that, the battery has no energy anymore, and then you can kind of send back that battery to recharge it again to, and in this case, for that ADP, which is the analogy for the which is the equivalent for that used battery, to be recharged again by the mitochondrion to make it ATP. Right, so those are the points of emphasis that I have for the nucleic acids video.